Hi, everyone. Can you hear me well? Okay. Yesterday, I was sitting there, and I couldn't really hear much. So just shout if you cannot hear. OK. Hi, everyone. My name is Dea, and I'm super excited to be here and grateful for the opportunity to talk about my work with you. Do I have any researchers in the audience, or scientists, or anyone just interested in healthcare? OK, an AI. All right. If I stood here a year ago, probably I wouldn't dare to put AI on the title page of my presentation, because machine learning was a bit cooler then. But everything has changed since the release of powerful chatbots that can maintain human-like conversation with us. And I'm sure that everyone here is familiar with large language models, I assume. Yeah. So when I was thinking about the topic, what to present today, I remembered one symposium that I've been to a few months ago, and there was a keynote that I listened to. I even have a picture of the presentation on my phone, because it was saying, healthcare is losing to football. Like, in what way? And then apparently, we are making better predictions in sports, and in football specifically, than in healthcare. And why is that? Because we have so much data. Most of the hospitals are digitalized now. We can sequence our DNA so cheaply and fast, and still, football is winning. So are we actually lagging in AI applications in personalized medicine, and why is that? I thought about it, and then I came up with a few reasons. And these reasons, of course, are by no means the exhaustive list of all the reasons why we are not advancing as fast as we wished for. But this is the list of challenges that I encounter day-to-day life in my research, and I will walk you through some of my projects and how I navigate through them to overcome these challenges. So the first one is that we are quite messy subjects to investigate, and so is our data. Just think about it. We are not grown in the lab in controlled environments. We all have different genetic backgrounds. We have different lifestyle and nutrition. And therefore, whatever we see in the data, sometimes it's so noisy. I've been working with patient data. It was so random. I swear, if I randomly generated numbers, I would see better patterns in them. So working with patient data is very humbling, let's say it like this. Then we have an issue of interdisciplinarity. In order to use AI tools in personalized medicine, you need to be fluent in several languages simultaneously. You need to be fluent in biochemistry, so the language of life. You need to be fluent in math and programming. And you also need to be fluent in law, because when it comes to patient data, you need to understand regulations. And ultimately, we all want to develop a drug, or we want to bring a medical device or a clinical assay to the market. And ultimately, you need to understand the regulations to bring this thing to the patient. Then we have, I would say, the most important issue for me as a scientist. We need to explain our models. We need to interpret our machine learning predictions. It doesn't matter how good it is. I want to know, I need to obtain biological insights out of it. Even if a model can predict something really well, just think about it. Why does it matter? If you have a drug and it works, and you know that it's going to treat you, but you have no idea about the mechanism of action, would you take it? Probably not, and FDA would never approve it. So you need to understand. And we are lacking here, because even though we have a whole new field of explainable AI, we have so many methods that can help us understand images and why AI makes predictions, it's still quite challenging to interpret them. And inherently, machine learning models were known to be black boxes. Now we are getting better at it, but it's still quite an important issue. And then we have privacy. And this is the key for why we are losing to football. Because sport data is quite easy to share. Everyone is interested. But when it comes to patients, the data is extremely sensitive. And there are some other layers of the problem. Because one, it's sensitive, and sometimes you need months to obtain the data. And then you have another issue with academics. We don't really like to share our data. There is a famous saying, if you ask an academic if they would share a toothbrush or their data, they would choose a toothbrush. Because first, you obtain the data, you spend so much time and resources on it, and I've heard one scientist saying, and it makes so much sense, that if they publish the data, someone can take the data, do some statistics on it, and then publish without understanding the limitations. Because again, coming back to the limitations, patient data is messy. It has so many limitations. You need to know them before you make any inferences from your data. So before we dive into the details, I will try to explain a few key terms that you might not have heard about from high school, I would say, but I will explain them to make sure that we are on the same page. So when it comes to language of life, but omics actually, we have 4 main fields and 4 main data types that we are dealing with. First, you have genomics. When it comes to DNA methylation patterns, we have the DNA breaks or structural variants. Then we have transcriptomics. To explain the main dogma, we have DNA, RNA, and proteins. So when DNA is transcribed, we have RNA-seq data, and then we have proteins that are translated. So this DNA information is translated into proteins. And we make up these very important molecules that facilitate biological processes in our bodies. And then we have metabolomics. These are mostly small molecules called metabolites that are produced by living organisms through biological processes. So my passion is proteomics. So I like proteins, and I deal with protein structures because the structure determines the function of the protein. And I work with biomarkers, so protein biomarkers. Biomarker is a measurable trait. It can be anything that helps us detect and evaluate a biological process. It can be clinical, like a symptom. It can be imaging biomarker, and it can be genetic. For example, a DNA mutation, certain type of mutation can be a biomarker as well. And biofluid, biochemical biomarkers. Those are molecules that circulate in our bloodstream, let's say, or the fluid that surrounds the brain, and we can measure these molecules and then diagnose, for example, or we can use them as a screening tool if our drug works or not. So biomarkers, that we have so many types, but I work with biochemical biomarkers for neurodegeneration. And when it comes to dementia, you've heard about dementia, right? So dementia is an umbrella term used to characterize various symptoms associated with the cognitive impairment. The biggest cause is Alzheimer's disease. Is there anyone affected somehow with Alzheimer's disease? Family members? Yeah. And what happens when they are diagnosed? Can you remember the first question that you have? So what now? We know that it's a progressing disease. It takes years to develop, and most of the time, the pathological processes start way early than we can detect them. And it's sort of a spectrum. So most of the dementia-causing diseases are overlapping, and their discriminative and early diagnosis is very, very difficult. But even after the diagnosis, we have no idea about the progression. When a patient is diagnosed, the key question that remains for them is that, well, what will happen to me? How many years do I have to live independently? And because dementia has such a heterogeneous disease course, even within the specific disease type, doctors cannot give a good indication of what will happen to this patient. So we cannot give a personalized prognosis. So one of the projects that I worked with was this, to try to understand or maybe discover novel protein biomarkers that can help us understand if a patient will progress faster or slower. So I wanted to use machine learning tools and try to use also proteomics data, proteomic profiles. So what does this mean? Nowadays, we can withdraw a small sample. CSF is a cerebrospinal fluid that surrounds the brain. So it's sort of like a window to the brain, let's say. And it surrounds the brain, so we know that it could be a good indicator of all the pathological processes that are going on in the brain. We withdraw them, and then we can analyze. And we have specific antibodies. Antibodies are also sort of proteins that are specific to other proteins, and they have DNA strands attached to them. Then they bind to it, it gets amplified, it has a unique code, barcode, and then it's translated back into the concentration, so normalized values. And this way, you can measure up to a couple of thousand proteins in one single sample for every patient. It's quite expensive now. I think it will get better at some point. But we had this proteomics data, and we also had their cognitive test scores. What this means is that it's a 30-point questionnaire. The higher the score, you have a better cognition. And you can see that over the years, everyone declines. Most of them decline. So our main thing, the first rate-limiting step is to cluster this data, because these labels do not exist. There is not such a thing as like a slow-progressing patient or a fast-progressing patient. You have to make it up based on the data. And making clearly distinct groups is quite challenging, because as I said, the data is quite messy. So you have these patients that overlap, and even the patients that are first grouped in slow-progressing patients, you can see that their follow-up is quite short. So if this patient passes away in a few years, even if they don't decline on the cognitive test scores, are they slow progressing? You never know. So you have to take them out of your data before you fit into your machine learning model. Because again, trash in, trash out. So you have to be very selective whatever you train your machine learning models on. Your models are as good as your data. And whenever we have the models, when we can make predictions, again, it's quite important to understand why we are making those predictions. So to interpret the model. And most of the time, there is a trade-off. So you have machine learning, shallow learning algorithms, then you have deep learning. And the further you go, the more layers you have, the deeper it is, the more difficult it is to interpret, to understand why the model makes those predictions. Sometimes you have to compromise the complexity for interpretability. Whatever we do, we need to obtain biological insights. So we need to know, for example, this is one single patient, and all these red, small things that you see, those are proteins that were measured. So you can understand which proteins contributed to this decision, and if this decision was actually correct. So this is a test data that we could analyze and see what are the proteins that are very commonly coming up into patients that are correctly classified. And whatever we do, whatever we obtain, all the biological insights, if we understand the model, it needs to be validated, because it's a patient data. Whatever we do, before we translate into a clinical assay, it needs to be seen again in other independent cohorts. And this is very challenging, because sometimes when we have new methods, they are high throughput, we can measure so many things, but those proteins that we find, they are novel. That means that they do not appear in lots of studies. We cannot really see those patterns in other studies. Those are just hand-picked. And that makes it difficult to validate. So you have to approach it in a different way. Let's say take one protein biomarker and then cluster your data based on that, and then sort of validate the other way around. So there are quite a lot of creativity involved in what you do afterwards. The case example 2 is that, let's say you have a good biomarker already. Even if it's a good biomarker, you need to measure it somehow. And I mentioned that we sometimes use antibodies. So these antibodies need to bind to your protein to detect it. And this binding is a crucial thing. This biomarker needs to be solvent accessible. That means that this biomarker, the antibody, should somehow get into it on the surface and bind to it. And it's not always clear how to measure the solvent accessibility. And most of the biomarkers that we work with are quite problematic. They are sticky. They tend to aggregate. And that makes them quite difficult to measure. Sometimes they are upregulated in CSF in patients with Alzheimer's disease. Sometimes they are downregulated, and we have conflicting studies. And we don't really understand quite often what's going on. So you need to have a good idea what's going on the surface to be able to detect it and measure it quite well. So this was one of the experimental setups that I used to investigate one of the good, promising biomarkers. It's also quite a problematic biomarker, because it's a brain-specific protein. It's called GFAP, and it can be measured in blood quite well, but it cannot be measured in the fluid that surrounds the brain, which is bizarre. We don't know why, because most of the proteins that are brain-specific, you can measure it in CSF quite well, and blood is just an indicator of what's going on there. And blood is quite difficult to measure as well. So why can we measure this protein in blood and not in CSF? Something interesting is going on. So I tried to investigate experimentally what's happening, and I used hydrogen deuterium exchange mass spectrometry. And what happens here is that you sort of incubate your protein in heavy water, and then you observe what are the parts of the molecules that are exchanged with heavy water, so deuterium. And then you measure that, and then you can sort of obtain an indication of which parts are more exposed in specific biological fluids. But this experimental data is quite difficult to interpret and understand still. And we need some computational tools to complement them, to understand them better. And recently, I mentioned protein structures. Just bringing you back to high school, you've heard about amino acids. So we have 20 different amino acids. It's like a language of proteins. And the sequence, the primary sequence, determines the structure of the protein. But there was a big biological mystery. We couldn't really predict well, based on a sequence, the structure of the protein. And recently, we made such a big progress. If you've heard about AlphaFold, that was developed by DeepMind, and they solved it partially, so now we can have much better predictions than we had before. And then we have another model developed by Meta, and this is a sequence of large language model, basically, but for proteins. So it's trained on the protein language, all the protein sequences that we have, and then they are masked. So let's say one letter is left out, and then the model learns, based on those sequences, the structural inferences. And this is a language model, so you can take the embeddings, it's very large, and you can fine-tune it for your specific biological questions, so they are quite useful. They give us so much insight. But before we go into those, you can just take your protein sequence, you can build your own machine learning model, and then predict whatever you are interested in. You can predict disorder, you can predict protein-protein interfaces, you can predict secondary structure, PTMs, aggregation propensity. I said that brain proteins are quite sticky. And then we have hydrophobic patches or solubility. You can have so many biological questions when it comes to proteins, and you can have your data and then just train your machine learning model on this data. But instead of building your own models, you can also use the existing ones now, the large language models that we have for proteins, and fine-tune them based on your data and make it better for your specific task, which is much more efficient and sometimes even better than building your own algorithm. So, based on this, we can now estimate protein structures. For example, AlphaFold can be used to predict the 3D structure of a protein that has not been crystallized, and then we can combine it with experimental data. So map it and see if it agrees, see where these exposure patterns fall, if they are conserved evolutionarily, and then you understand much more than what you would understand based on just experiments. And then you can mix and match all the other properties as well, where on the sequence, so this is just a sequence following, and then you can have different criteria where exactly this protein sequence matches. Let's say the disorder or secondary structure or evolutionary conservation. So, how do I actually navigate through the challenges that we have in personalized medicine? I try to stay critical, as critical as I can, and then whatever I find, just validate and try to see it in other studies. When it comes to interdisciplinarity, just be open to different ideas, talk to as many people as you can, because everyone has insights. Even if someone is just in different field that you never thought that it would be useful for you, it can be very useful. They have usually quite interesting ideas, and they have a bigger picture usually. Sometimes you dive into the detail and you don't see what's going on. Then when it comes to interpretability, sometimes you have to compromise the complexity of your model to be able to interpret your model and your predictions. And when it comes to data and privacy, sometimes we cannot do anything. Sometimes I get requests to share the data, but I just can't, because you need to wait for months. You have to sign agreements to get them. But whenever we make computational tools, whenever we develop something, we always publish on GitHub, and we also publish scientific papers, so anyone can actually see whatever we are publishing. And I try to be open. All the decision making that is used for data cleaning, data curation, and machine learning models, all the specifics are published. So anyone can have a look at it and tell you it's peer reviewed. So people examine that and they tell you if something is wrong, or you can have done something better and try something better. And we just explore everything, and then we validate, and then it's translated into the knowledge at some point. So talking about interdisciplinarity and connecting and communicating with each other, let's connect. And thanks a lot for your attention.